Sibley Lake's own nursery rhyme. Was Jack and Jill as happy as we thought? Let's find out. On August 29th, a missing persons report for Jack Smith was filed by his wife, Jill Smith. Earlier that morning, Jack had gone out for a fishing trip, while his wife supposedly had gone to Shreveport for a shopping spree. While the investigation was going on, police noticed that Jill had grass on her pants, so they took that as a sample. Later on that day, they found Jack's boat and his body at the bottom of Lake Sibley. On the boat, there was way too much blood for it to have been an accident. After they pulled Jack's body out from the lake, they found grass on his pants. They took both samples to professional geneticists so that they could find out if they were matching. Was Jill out for blood? Let's find out.
Okay, to begin, the professional geneticists had to extract DNA from both of our grass samples. This DNA was too tiny for us to do anything with, so we had to amplify it using PCR, which stands for polymerase chain reaction. This amplified DNA was then separated using AGE, which stands for agarose gel electrophoresis, into big and small pieces of DNA. The big DNA pieces move slower than the small pieces. Before cloning, we had to make sure that the DNA was not contaminated, so we purified the construct. During cloning, our vector, which was our transformed E. coli cells, strand JM109, had our amplicon inserted into it. This allowed for it to incubate and to further grow into separate colonies. After the different base pairs were dyed with the big dye into the 4 different colors, it was put into the sequencer and mapped out in the order of the base pairs so we can figure out if our 2 plants matched from Jack and Jill. Our word for the DNA that was amplified is just amplicon. The vector used, like I said earlier, was the transformed E. coli cells. The strain mentioned before was JM109, with our forward and reverse primer being 1A and S64.
Okay, so after the sequence was, after the DNA was sequenced, our sequencer provided a text file and a data file showing the sequence of the DNA in a colorful electrophoregram. The reason why the sequence was able to see the DNA is because each nucleotide was dyed in a different color, different fluorescent color. And so after we did that, we plugged our data into the Gen bank. Put the sequence into the Gen bank to find the match for the 2 samples that we got from the grass. And our sequence was blasted, and we got that the grass that was on Jack was a Lolium multiflorum. It was 100% match in the database, and then the one that was on Jill was the Dactylactinium egyptium, which was also 100% match in the database. They are 2 different, completely different samples.
Considering that the DNA from the 2 samples were not a match, investigators could not tie Jill to the scene of the crime or to Jack's death. Because of the amount of blood found on Jack's boat and the severity of his head injuries, it cannot be concluded that his death was an accident. Therefore, police could open up a search to find the murder weapon, which could lead to the killer and who may very well be Jill Smith.
Did Jack fall down and break his crown? Was Jill out for blood? Further investigation is needed. On this next episode of Who Done It?
The only